Yeah, my name is Eric, and I'm going to talk about something completely different. I'm not going to talk about my work because I just got here 2 months ago, maybe for a future one. What I'm going to talk about is two things I'm really interested in, and that's immune systems and crabs, which sounds really funny when I say it out loud. So in humans, there's two types of immunity, two branches, adaptive immunity and innate immunity. Adaptive immunity is characterized by B cells and T cells. It's long term, takes many days, and it makes antibodies, which everybody's familiar with. What I'm going to talk about is innate immunity, which is much faster and has different types of cells, including barriers, physical barriers. And so I want to tell you about innate immunity in the horseshoe crab and how it benefits every single person in the room. So horseshoe crab, a very simple creature, you've probably seen it on the shore somewhere. It's got these legs. It's actually more closely related to spiders and scorpions than it is crabs. Legs here, book gills, and two eyes on top of a hard shell. And so it looks very ancient, and that's because it is very, very ancient. It comes from about 450 million years ago. This predates all dinosaurs and has survived multiple mass extinctions. And so when it was around at that time, the Earth was a lot different. So this is what the Earth looked like around 450 million years ago, with a large Gondwana supercontinent on the bottom and these smaller continents here. And so life on Earth at that point in time was dominated in the oceans, and so it was teeming with life, and it looked very different. So you had many organisms here, and on land it was very oxygen poor, so there was no animals there. And down here in the oceans, there wasn't any even jawed fish. They came 100 million years later, and let me tell you, the jaw was the must-have device of the Mesozoic. And so the crab is really old. So how did it survive for such a long time? And there's a lot of reasons, but one of them is an immune system that's very impressive. So the white blood cells of the crab has this compound that when it encounters bacteria, it forms this gel-like substance almost instantaneously and surrounds and traps its pathogens. And so this thing is used by the FDA to test any drug and any device that goes into humans without the digestive tract. So how do we get it? We catch crabs on the surface by hand, we clean them, we put them in this Game of Thrones type contraption here, and we bleed them. And their blood is blue because copper transmits the oxygen instead of iron. And they survive this process. We throw them back in the water. The blood is blue, we spin it down, and we get this dried compound, limulus amoebocyte lysate, that then is used to test in extremely small amounts of bacteria and all of these things that the FDA tests. So how does it do it? 20-second molecular biology here. So gram-negative bacteria encounters the cells. The cells release tons of factors. This initiates a cascade of events that ends up with coagulin essentially gelling around the intruder and isolating it when it's eventually cleared by the organism, the horseshoe crab. And it's called a cascade, and these things are very common in biology and immunology because one event leads to another. And in every one of us right now, there's a ancestor or a descendant of this pathway called the complement pathway that's protecting us right now from bacteria. And so this is just what has come 450 million years later. So this even has applications in space because it's so simple and so sensitive. They use it to protect astronauts. So shown here is the lab on a chip applications development portable testing system, and so you inject a sample in there and it tells you if there's any bacterial contamination, and so they use it to protect the astronauts and to protect space. My 4-year-old daughter thought this was really cool, so she drew a picture of a crab with a sword beating up a germ in space with the sun and the moon, Saturn and Earth, and of course the space butterfly. So it's not all sunshine and space butterflies for the crab. There are many modern threats to its survival, as you might imagine, with a lot of animals. So very early on, before synthetic fertilizers, they used to grind up tons of horseshoe crabs and use them as fertilizer. It's a very, very good fertilizer. They also feed poultry with it. And now, even today, they use it as bait to catch conch and eels. It's a very good bait to catch these animals. And so even though the bait and fertilizer industries fisheries have started to use less and less crabs as you go over. The biomedical harvest has been increasing over time. But they survive, Eric. Why do we care? They're okay. Well, people have been noticing a lack of females on the beaches during the spawning of the horseshoe crab. So it's become more or less kind of a sausage party, which people want to figure out why that is. So a group set out to determine what the sublethal effects are of doing this bleeding. And so what they did is they essentially attached pedometer type things to 50 different female crabs, or had them run around in a wheel made of a 5-gallon buckets and PVC to track how much they move. And what they found was if you bleed an animal, compare it to a control animal that wasn't bled, what you see is they start to be more active on a 24-hour scale as opposed to a 12-hour scale, which corresponds to the tides. And these animals need to correspond to the tides because that's when they come in and out to do their breeding. And so it gets a little bit better when you look outside, and not with this lab wheel inside a lab. They do a little bit better, they recover a little bit faster, but even if you bleed them and they're outside, they start to lose this circatidal rhythm, which is very important. Another way to look at this is to look at just percent activity over time. So how active are the crabs compared to ones that are bled and ones that aren't bled. And so you can see that at about the time that they're bled, they're about equal, maybe a little bit more active. But over time you get about 50% of the activity in those that have been bled, which makes sense. They're taking quite a bit of blood out of these animals, probably just enough so they don't die. And so this study and some more recent studies have suggested that it could be this bleeding that has led to the paucity of these crabs on the shore. So an effective innate immune system goes a really long way in the survival of a species. Extract from these horseshoes amoebocytes, the blood cells, is exquisitely sensitive to pathogenic invaders, and bleeding these crabs, if it doesn't kill them, may impede their survival.